But by a landslide, the worst deal of the summer, as voted for all by all these agents that the Athletics spoke to, was Benjamin Sesko. Their top 5 were this. Facundo Bernardi, who went to Chelsea, that was their 5th worst deal. Number 4th worst deal, Jadon Sancho. Number 3, the 3rd worst deal of the summer was Wissa. And the 2nd worst deal was James Trafford to Manchester City. Isak and Wirtz got 1 vote. Anthony to Betis got 1 vote. Gittins to Chelsea got 1 vote. Connor Cody, who I don't even know where he went to, got 1 vote. But Benjamin Sesko got 6. I mean, he's literally not started games until recently, and then he scored 2 goals in 2 games in the Premier League, but he is the worst deal of the summer. I mean, tell me Man United don't boil people's piss without telling.
So, good afternoon. Once again, you are welcome on board. This is another opportunity for us to continue the second part of this presentation that we have to do on instrumentation for microbiology and environmental biology. Don't forget that in the previous presentation, we made mention about what instrumentation is all about and where we define instrumentation as the scientific foundation for observation and measurement. So we also went as far as defining instrumentation as the major bridge between environmental biology and the knowledge gap. So that is to tell you that how important instrumentation is in the real sense of it. And in the previous presentation, we look at the learning objective that one of the objectives of this presentation is that students should be able to define instrumentation, be able to identify key instruments used in biological studies and explain how measurements are obtained and understand. We'll be able to understand the importance of accuracy, specificity, and so a few. And we have actually given example of instrumentation. Then the component of instrumentation, we find time to look at that, and we divided into 4. That is the sensor, the signal conditioner, the display recorder, as well as a controller. And the principle of measurement. So that is whatever might be your readings or your actual measurement, you have to compare it with the standard. And we look at the types of measurements parameters. So we also look at manual versus automated, in which I actually gave that in our comment section, the community section, and whereby people, some of you actually got it right, giving out example, the diagrams of the photograph of manual versus automated, and I can see that actually most of you actually got the question very, very correctly. Then we'll also look at accuracy, precision, and sensitivity. That these are very, very important when we are talking about instrumentation, because you'll be able to detect very small changes in measurement. So we also look at data acquisition and processing, and we talk about calibration. For example, we look at a pH meter that is being calibrated using buffer 4 solution of a pH value 4, 7, and 10. And we actually talk about why it has to be. Then also, we also said that you could expect errors in measurement. And when we talk about error in measurement, it could be as a result of error from human mistake. It could be faulty instrument, or it could also be environmental interference. And we also look at it that the key into this is a regular maintenance and calibration. in order to be able to reduce or minimize this error. Okay, we said maintenance and care is key in maintaining accurate reading, specific reading, and so on and so forth. So, safety instrumentation, we look at part of how you can maintain, how you can be safe when it comes to instrumentation. Is your personal wearing of personal protective equipment like hand glove, like your lab coats, so on, so that you will not get contaminated and you will not also introduce contaminants into whatever you are doing. So, for example, somebody who is handling very contagious or infectious agents, you should be very, very careful not to contaminate yourself, infect yourself. Then, at the same time, you must not even allow contamination to get into what have you. Example of instruments, we look at that, and we also look at a summary of this. So, definition, what are the scope and definition of this? We look at that, and once again, we also talk about instrumentation combine scientific principle with technology that has to observe the physical, chemical, and biological phenomena accurately. So we look at historical development. The early scientists used instrumentation like thermometers and weighing balances. The scope, we define it into different areas that you have laboratories, research laboratory, environmental monitoring station, hospital, industries, and so on. Then branches of instrumentation that is into analytical processing, uh, biomedical and environmental instrumentation. And each of these are specialized for particular application. So we look at analytical instruments, process instruments, biomedical instruments, as well as environmental instruments. So also industrial. So now that was the area which we wanted to discuss about was emerging areas whereby we talk about innovation, including biosensors, AI integrated monitoring system, and so on and so forth. So today we are going to look at instrumentation in education. So how important is instrumentation in our education? Practical knowledge of instruments. Definitely we prepare students for laboratory and industrial roles, improving our employability and technical competence. So what we are saying in essence is if you are very good in handling equipment, practicals, handling and all this one, that will increase the chances of you getting employed than somebody who doesn't have that experiment. Somebody who can, for example, don't forget that in our secondary school days, we were introduced to titration, we were introduced to qualitative and quantitative analysis. And if you are able to get very accurate record of whatever the scientific readings, so people will like to employ you than somebody who doesn't have it. So our instrumentation, your knowledge in practicals will prepare you whether you are going to be gainfully employed or maybe at the end of the day you might not even get to be employed at all. So if you want to get ready-made job at the end of the day, make sure you increase and you improve on your practical knowledge, especially manipulation of the use of instrument to do a lot of things. For example, if you want to diagnose, if you want to test, if you want to do a lot of things, do you know how to handle PCR? Do you know how to handle microscope? Do you know what? What do you know at the end of the day? So that is the usage or the relevance of instrumentation in our future career. So then it also has a very good role. Thank God this slide is actually talking about data science. Data science, in which I said some of you should actually register for because you are going to start talking about. I told you this is the era of artificial intelligence. It is a digital era, and if you want to continue to be relevant, That means you must be able to understand the language of computers, the language that internet will also understand. So what we are saying in essence is, I told you in the afternoon that the relevance of what I asked you to register for might not be immediate, but later on you are going to benefit at the end of the day, including even giving you additional job opportunities. So nowadays, the previous slide is talking about your improving, your knowledge in instrumentation is increasing your employability level, your technical competence. So somebody who knows computer, how to use computer, and somebody who does not even know how to use computer, he doesn't know how to solve the internet, how to search for information, how can you, what language. Don't forget that we say computer does not understand any other language than zeros and one. So that means if you are going to know, if you are going to be trained in any usage of any instrument at all, that means your level in data science, you should be able to integrate it. You can see instrumentation provide accurate real-time data that supports modeling, simulation, and predictive analysis in life sciences. So what I'm saying in essence is that data science, programming like Python, and some others, SQL, and some others like that, they are one of the hottest hot cakes in biological science or in life sciences till today, because your knowledge, if you know how to make use of Python or Java. In fact, Java is even becoming so outdated. If you are so good in data science, honestly speaking, let me just tell you that you will just be at the four corner of your house and you'll be getting job from all over the country. So instrumentation, how can you integrate data science into your instrumentation? So this involve you making use of this data when you after you must have used your instrument, calibrated instrument, to get a lot of readings. You get a lot of data. You see complex data coming out. So if you don't know how to utilize that data, your homework is that you just be in charge of microscope. You just be supplying them with the results, readings, and you don't know how to manipulate this data. You don't know how to convert this data in order to be able to make informed consent and be able to make certain policy about treatment, about drug to use, about so many things. At the end of the day, you just discover that somebody elsewhere. That is why it is very good for you. The training on data science is coming at the real time that you will actually need it. So what are the limitations and challenges in instrumentation? Number one, high equipment costs. Now, let me tell you something. Now, do you know? Okay, let me just tell you something. In those days, for some of us that we eventually get to know PCR at the end of the day, PCR then, it was only, let me say, throughout this country, it was only uh, about two or three places whereby you can see PCR machine. Now, as at that time, even if you want to make use of a PCR, sometimes you will be working on PCR, just cocktail, one cocktail, or let me just say one preparation. Let me use it preparation at this level, but one cocktail of PCR reagents, when you quantify it, is something that should be more than 25,000 naira at a go. And sometimes you can even have more than 25,000 naira cocktail at a go. And by the time you now run it, you are going to run it for more than 96 hours. And after 96 hours, that is about more than 3 days, more than 3, 4 days, around 4 days now. One thing you run for 4 days, and at the end of the day, you now discover that you are not getting accurate records. You are not getting what you're supposed to get. So at the end of the day, it is tiring. So, and equipment, just one that equipment, you know how much it costs. In fact, a sequencing machine that I saw it in one teaching hospital. When I saw it and they brought out one of the spare parts of that equipment, one of it cost more than $90,000. So to actually tell you that high equipment cost, then need for skilled personnel, somebody that can handle it, is a major challenge. Now, when you talk about equipment, how many institution, how many hospital that can actually that can acquire um an equipment that one of its parts worth more than $90,000? How many? Now, skilled personnel, there are so many people. You have some equipment, some places that you do not, they don't even know how to make use of it. Then calibration error is very, very important. So we can have people that can, so there are some people that you have the equipment, there are no trained personnel, and sometimes if you don't know how to calibrate the error, you just discover that this thing is not working, and you want to troubleshoot, you make a lot of calls, you don't have and at the end of the day you will just go and abandon millions of dollars of equipment just because of what you you were able to afford the cost. You are skilled in terms of personal training, and then sometimes it's not because of your calibration error. So then sometimes maintenance difficulties, oh, especially in this Africa where you are not so sure of a regular supply of power. You know what it means. I know in part of this country there are places whereby to even get light, you'll be thinking about 2 3 months that there's no light in some places. So especially in resource limited settings. So these are part of the challenges. So just like I said, what are the importance of instrumentation in life sciences? We are talking about life sciences. Life sciences are basically biological sciences. So that actually deals with life, that handles life. Biochemistry is also involved, biostatistics, anything that has to, any science that involve or contain bio, uh, that means what is known as life sciences. So instrumentation plays a very crucial role in observing, measuring, and controlling biological and environmental processes. So it enables scientists to produce accurate and reproducible data, making discoveries more relevant and more reliable. So what we are saying in essence is in science, if your instrumentation knowledge is lacking, you just discover that there won't be any new discovery. And when there is no new discovery in your area, for example, now I can tell you that the anti-TB vaccine that we have been using, BCG, is more than 100 years old, and since that time we don't have it. So what we are saying in essence is just that if there is problem in instrumentation, there won't be anything, no accurate record, your record are not reproducible, or you get a different result with the same procedure this morning, and after some time when you want to repeat it, you are getting a different thing. That is unreliability of your data, and which is not going to go well in the um so life sciences rely on precise data collection. I told you the life sciences will need to collect data that are very, very precise, that are specific, that are accurate. So in order to be able to translate all these data into biological signals, into measurable values that help to understand processes like microbial growth, enzyme activities, and environmental changes. So what we are saying in essence is that we actually need accurate record. We actually need instrumentation in order to be able to get and keep accurate record, to track accurate data, especially when it comes to a lot of environmental changes. So accuracy in observation. Scientific research requires quantitative data. Scientific research requires quantitative data, and instruments minimize human bias, allowing research to measure phenomena such as pH, turbidity, and temperature with precision. So, in whatever measurement, in whatever experiment you are carrying out, it is very, very important for you to not to play with the instrumentation. So you need to know that. Then research and discovery. From the discovery of DNA double helix, I told you in the class we had today that anything in biological sciences, if you cannot understand the molecular structure of the DNA, you just discover that you are nowhere you are nowhere to be found in the field of biological science. So from the discovery of double helix DNA in 1953, using X-ray diffraction to the visualization of cell through electron microscopy. So instrumentation has driven major breakthrough in biology. So what we are saying in essence is you go back to most of the giant discovery in biological sciences, it was as a result of good knowledge and in instrumentation, so which is very, very important. So medical and clinical application, clinical laboratory depend on instruments such as a spectrophotometer, ELISA, the one that will measure the antigen antibody reaction, that is ELISA. PCR machine, I just mentioned it. This is a machine that will amplify. You just have a fraction of DNA, and you need this fraction of DNA to be amplified so that you'll be able to sequence it and be able to produce it. So what we are saying in essence is PCR machine is very, very important in diagnosis. The same thing with spectrophotometer, the same thing with ELISA and other equipment that they use to diagnose diseases as well as analyzing biological samples. So environmental monitoring. Instrumentation helps monitor air and water pollution, soil composition, and microbial contamination. Devices such as gas analyzers and dissolved oxygen meter ensure ecosystem. So industrial quality control. Instruments ensure standardization of pharmaceutical, food and water products in order to be able to detect impurities, measure concentrations, and validate product safety. So what we are saying in essence is instrumentation is very, very relevant in industries. And industry like pharmaceutical, they depend mostly on their equipment in order to get accurate record about them detecting impurities, measuring concentration, and also validating product safety in what might be their product. So it is very relevant in industry. Now, what do we mean by automation and efficiency? Don't forget that we have discussed automation versus manual ever before, that automation will require less human effort. So automated instruments improve speed but reduce human errors. Examples of automated machine include autoanalyzers and programmable spectrophotometer used in continuous monitoring system. Now, let me just burst your brain. Honestly speaking, if there is any time you need to actually appreciate what data science is all about, it is now, because it is going to give you an edge over somebody who does not have that knowledge. So what I'm saying in essence is just that you should just get yourself prepared that there is nothing you can do in the world, in this world, and the world to come in the next few decades if you don't have digital knowledge, if you don't know how to command computer, how to run certain programs on computer. If you don't know them, honestly speaking, it might be so difficult for you. So what I'm saying in essence is just that we need to know, we need to know and appreciate the importance of data science, especially the program that is coming up, that is 30 days, 30 minutes for 30 days challenge. So it might not be too useful for you now immediately, but you are going to need it later on. Later on, even if you want to continue with your education, you are going to need it. Apart from that, there are other side job that you can do, which we will see when we get there. So what we are saying in essence is automation. If your machine is automated and human that is supposed to automate that machine is not automated, you know how unfit you are going to be. So what we are saying in essence is just that the knowledge about data science is going to help you because you are already automated, you are already inclined, you are already moving into data, digital world. So that is to actually tell you. And the more automated you become, the more efficient you become, so to say. So the more you understand the language of the computer, the more efficient you become, the more relevant you become, the more useful you become, and the more chances of you getting, making money and getting employment. So what we are saying in essence is just that instrumentation, whatever knowledge you have nowadays, you should be able to integrate digital digitalization with it, digital world. So that is just a challenge. That's why I said it's going to be a challenge. If you can spend 30 minutes for 30 days, let us see where you are going to be. Let us see whether you are not going to you are not going to stand a better chance than somebody who does not even know anything about, even if your phone is misbehaving. Sometimes we don't even know what to do than just, even if you want to send ordinary mail, there are some of us that would not even know. You want to open Gmail, you don't even know. But with knowledge and in automated your automated knowledge, you'll be able to practically, even if you have not laid your hand on that machine before, because you have automated knowledge, by the time you get there, they'll just you just say, oh no, don't worry, don't worry, it's something I can handle, just give me the manual, or let me just go to the internet, just go and go to YouTube, type the name of that machine, you just see it, you watch it, and then before you know it, you know how to handle it. But the question is, if you don't know how to move from one place to another, especially the use of YouTube or social media, that means you might not even know, you might not even be relevant in some other time to come. Please, I want you to seize that opportunity, seize it as an opportunity that you might not have that opportunity once again. I just told your rep, I only ask your rep very simple thing. Go to Google, just go and type how much is it going to cost you for you to have basic, just ordinary basic knowledge in data science, that is Python. So at the end of the day, you will see that and don't forget that so many people will not know about opportunity unless that opportunity is found. So let us just go back to this. What I'm trying to say in this slide is that the more automated our instrumentation is, or our instrument is, the better the efficiency and the chance or chances of it reducing human error. Okay, then data storage and analysis. Oh my God. So modern instruments store and process data electronically. So if you can still see why I'm sounding it now. If you are not into data science, you won't know how to handle modern equipment. You won't know it. So you can see it here. Modern instruments store and process data electronically, enabling easy retrieval, analysis and integration with bioinformatics tools for deeper insight. So thank God this is actually telling you all this now. Second semester is when you will offer a course which is about informatics. But from what we have seen so far, just like I said, you can see data. Everything nowadays is about data. Data, documentation. We have too many data, too large data that we don't even know how to make use of them. So there are so many people, there are some industries that they are becoming folded. They are folding up huh? Because of what? They have data, but they don't know how to analyze that data. That's the reason why that if there's something you need to pay for, if there's nothing you need to be looking for in any area, you could be, even if you are selling fish, if you are whatever. See, it is high time you become automated. It is high time you become a digital person. Our phone, nobody is actually talking about pressing phone again or what do we touching phone again. Android already. So that is that. Then teaching and training. Students trained in instrumentation understand both theoretical and applied aspect of science. And hands-on learning enhances problem solving and analytical skills. What do we mean by hands-on? Hands-on is like for example now, the practical you can handle by your own, something you can see and you practice. By the time we start that data science, it is going to be hands-on. Hands-on in the sense that after we must I might have the lecture is not going to be more than 30 minutes because it is 30 minutes for 30 days. So it's not going to be 30 minutes more than, I mean, in a day. So the next thing, after you must have gone, you yourself, you need to train your hand. You need to exercise your hand. You need to, for example, now, if you are being taught how to handle microscope and it's something we have just demonstrated, or we are reading out a manual for you, reading procedures, do this, do that, do that. The one you yourself, is not that something that you watch your friend doing, something that you yourself you can lay your hand on is what is called hands-on learning, which enhances problem solving and improve your analytical skills. So, for example, somebody that handle microscope um is going to solve more problem, is going to be more knowledgeable, is going to have more skills than somebody who does not know how to handle it. So, just like I said, this is the world of data. Everything in science is giving us data. Look at, for example, now, if we want to actually check the, since the beginning of biological sciences, can we have a document? Can we have data about everybody? All microbiology students, their including their name, their age, uh, their year of graduation and everything. Where are they? Their projects, where are they? The data they generated from their projects, how are they handling it? So what we are saying in essence is that you should always strive you should always strive to be trained on how to make use of certain instruments so as to be able to excel well in biology. I've really discussed a lot about, I mean, for you in today's class we had. So then economic importance. So instrumentation supports health, industry and environmental sector, contributing significantly to national development and job creation. So what we are saying in essence is just that as far back as the year 2000, we discovered we discovered how much people and how much in terms of dollars that US is making from instrumentation, their own equipment. They are able to, for example, now if you now have just let us imagine now. Let us say that you have a very small room. Oh. That small room, you have a microscope, you have a pH meter, you have a PCR, and let's say at the end of the day, let's say you have just 1 million to set it up. Let me just say that. Let me tell you one thing. Within the shortest period of time, within the first year, the money you make, you will make will be times two of the money you have uh, actually invested. So what I'm saying is very simple. Any hospital whereby they can carry out a lot of tests, medical tests, X-ray, diagnosis, ELISA. So go and tell them, go and check. You just discover that the more equipment they have, the more accurate their data is, the more money they will have. So it is going to support job creation, it's going to support them economically. And before you know it, just see that it's because of the analysis, it's because of the instrumentation or the instrument that they have. And that's the only thing that is putting them on world map. So what we are saying in essence is one of the reasons why some people, some youths, some people, why they are still very relevant today is because of their knowledge in instrumentation, in handling some instruments, because of their knowledge in automated machine. Don't let us just miss word about that. So that is that. So then in the area of disease and detection is very, very important. So don't forget that during the era of COVID-19, that time, if any hospital or any establishment had PCR machine, PCR machine, number one is that you do and check how much they were making in terms of dollars. So what we are saying in essence is just that you as a biological scientist, as relevant as you are, the more you want your name, the more you want to make it in life, the more should be your knowledge in instrumentation. So glass blowing, I want to during the time, I think some years ago, that glass blowing, we only had about three or four of them in the whole of West Africa. So now imagine if how much they will have made if we have people that can actually go into that. So what we are saying in essence is that some of us, instead of us to go for teaching, instead of us to go for lecturing, or some of us to go into a lot of places looking for money, and you want to make money, just go and get yourself trained in handling of equipment, handling of sample. I know how much I paid for what is called whole genome sequencing. Whole genome sequencing that you just pick up a microorganism or bacteria that you want to know the alphabet, how many alphabet and the order of that alphabet. It's something that you will be paying just for one sample, something that will cost you more than 500,000 just for one sample alone. So what we are saying in essence is that our knowledge in instrumentation go a long way to be able to tell us whether we are going to be successful in biological sciences or life sciences, so to say. So sustainability and environmental impact. So by providing data on waste, you can see everything data. Everything is about data these days. So waste, if you can have good utilization of waste from data, data from waste, that of pollution, that of ecosystem changes, instrument aid sustainable practices and promote environmental conservation. So what we are saying in essence is that once you have instruments that will be able to tell you about waste management, be able to tell you how polluted your air is, how your ecosystem changes. If you are able to analyze that data and you have it, utilize it, you know how many or how much the environment is going to be so stable and how you are going to even sustain. So what are the future prospects? The future prospect of instrumentation. So number one, the future prospects, that is instrumentation of today and tomorrow. That is another way of understanding what we call future prospect. That is, what are the chances of survival? What are the chances that in the next few years you are still going to be relevant? So honestly speaking, if you want to be relevant, the only thing I will tell you the only thing I will tell you is integration of what is called nanotechnology. If you are not going to nanotechnology, you can go for robotics or you go for artificial intelligence. And whether it is nanotechnology, robotics and artificial intelligence, if you do not have knowledge about data, that means you are not going to be relevant. So that's why if somebody want to predict how the future will be, a time is coming. It is already here. Don't forget that before COVID-19, there was nothing. Even if there was something, the issue of what is called virtual classes was not that relevant, was not that much. But nowadays, and that is where everything is moving nowadays. Now there's something we now call a net. Net, if you want to go for ordinary NYC, if you don't register in net, that if they don't have your data, if you are a student, if you are a lecturer, whosoever you may be, whether past on the in the whether past or now, if your data is not in net, let me tell you, there's nothing you can do in this country. Nothing you can do in this country. Now, one other thing to tell you about the artificial intelligence. Nowadays, so many of you knows how to use the artificial intelligence, but there is something we call agentic uh, robotics. That is, you are the one, instead of you to depend, instead of you to start using artificial intelligence, you are creating your own AI software. So it's still very, very possible. You might be in microbiology, you might be a biological science student, and if you don't know how to make use of all this, to integrate them together, to be able to know about data, how to manage data, how to utilize data, how to keep data, how to manage data, and how to use data to make decision, honestly. So it will revolutionize life sciences. By the time it revolutionize life sciences, it's going to lead to smarter automated analytical system. So what we are saying in essence is just that the era of what is called Gen Z era now is a digital era. And if you want to become so relevant, you need to make sure that that is a part of what you have to do. So instrumentation has transformed and is still going to transform biological phenomena into measurable. Gone were those days that we just continue to measure photosynthesis on in the flask. Want to do photosynthesis, get all this equipment, get all this. Let me tell you something. If you want to know what happens in instrumentation, it is just a matter of what is called simulation. You simulate it. What is you use your knowledge, digital knowledge now, to simulate, to imitate what is going to happen. So what we are saying is, you know that nowadays, vaccine production or vaccine design nowadays is no longer going through all the traditional processes that is going to take like 20, 15 years. It is now when you have your computer system and you have your own data source of internet, you will design what is going to happen. We will predict what is going to happen when that is where you design your vaccine. When people take that vaccine, you will simulate the immune system. You will see the same way it is going to react in human body on your computer system. And it's not because of anything, it is because this is the world of data. It is the world of digital or digitalization. So, and that is why the backbone of biological sciences nowadays will be something that is going to be based on how we can utilize instrumentation. But some of you might not, when we are talking about instrumentation, you won't know that even computer is an instrument. Oh. Computer is an instrument, is an equipment that you can use. In fact, and that is the modern day laboratory. Let me tell you, that is the modern day laboratory where you can do a lot of things. You might not visit the laboratory, and what is going to happen in the laboratory, is something you can even know, is something you can even discover, is something you can even practice with your computer system right away from your room. You'll be able to predict what is going to happen in the future, what's going to happen about antimicrobial resistance. You design, you simulate it. So that is why this era is different. It's a different era. It is era of digital, and just like I told you, this is going to remain the backbone of modern research. It's going to remain the backbone of innovation. It is going to remain the backbone of our education in life sciences. So you can nobody, if anybody is just deceiving you, telling you that you are wasting a lot of money on your data on internet on this one, that person does not want you to know, does not want you to be relevant. The person is not wanting you to be employable. Is not wanting you to develop your skill. So what I'm saying in essence is just that instrumentation, as we can see it, is nothing but something that is going to be the backbone. It is the backbone. It started from somewhere and it is now somewhere. This is the era of automation. This is the era of digitalization. And in the next future, it is going to still remain like that. And what have you. So once again, I really want, if you have not registered for that Python, it is what you should register for. It's what you should register for. It's going to prepare you for the future. Not only that, it's still going to make you to be able to for side hustle. Now you want to post ordinary picture, and you want it to trend. There are certain commands, there are certain command language that you can make, that you can use. So that if you don't know how to use all this language that computer will understand, at the end of the day, you just discover that it's going to be a waste of time. Now let me tell you that if you want to carry out a photosynthesis, you want to see what microbiology is all about, what human system is going to be, digestion, all these processes. If you don't understand anything at all, just use your own method of automated automation, and you get to see them and see them live. You simulate, you imitate it, you'll be able to watch it live. So this is the era whereby what you want to give to them outside, the product that you want to produce for people, you you must have seen it before it is going to become acceptable to you. Don't forget what I've said today, that instrumentation transform biological phenomena into measurable, not only measurable, that is something you can measure, actionable data. Action is not ordinary data. Time has gone when what you just see is just that you just have data without anything to utilize it. It's not going to do anything. So don't forget to register for my course. It's going to be registration is free, and attendance for you to qualify for certificate is going to be more than 80% in attendance. So don't forget that it is a 30 days, 30 minutes for 30 days challenge between November 1 and December 1. And it's not only you, somebody who is not even in science at all can even join as well. So you have the link. I want to see you in the next video. So once again, um, it is good to have you now. So I want you to comment on in the comment section. I will be looking for your comments. Comment section. Okay. Any question? Please, question and answer time. Question and answer time. Okay. So let me ask you just two questions. Let me ask you two questions. Okay. How can you integrate automation with instrumentation? Okay. Yeah. How can you any response? Integration of automation with instrumentation from our presentation today. How can you integrate automation with instrumentation? Any response? Any response? Okay, then I want you to somebody to summarize. Summarize today's lecture in 300 words. Okay. Automation uses data from instruments to automatically control processes, keeping operations stable and efficient. Okay, but how can you integrate it? Integration. Okay, I understand. I understand. Rabi Europe keep. Okay, that is a bit um to the answer. A bit to the answer. So without wasting our time, I want you to attempt the question. The question I just gave now. Number one, automation versus how can you integrate automation and instrumentation together? How can you integrate it? Then the second one is okay, PCR, polymerase. That's good. Polymerase chains reaction. PCR, that is polymerase chain reaction. So polymerase is an enzyme. Polymerase is an enzyme. So it's going to amplify. For example, polymerase chain reaction is a machine that when you just get a very small fraction of DNA, for example, let us see that maybe somebody committed a crime, and the only thing you can recover from that is you want to carry out the forensic analysis to know whether somebody who is a suspect was actually the person, that is the right person that was accused. So what you do is just you collect a small sample of that soil, you collect small sample, then you go and analyze the DNA from the soil. When you do the DNA and you extract the DNA, then it's going to be a very small fraction. So you now amplify it, and by the time you amplify, so you now need um, polymerase chain reaction machine to be able to analyze that. So we are going to, don't worry, we are going to have a time whereby we will know the detail, we will learn detail about PCR, the mechanism, how does it work, how can we read it, and what should be the relevance of it whenever I want to. But don't forget, PCR does not make mistakes. It does not make mistake. It is a molecular base, and anything that has to do with molecular, that is with DNA, is more specific, is going to be more accurate than you. For example, now under the microscope, you may, one way or the other, you may diagnose that, you may identify that this one, you might not be sure. You may just say, oh, this person is suffering from this type of infection. But when you are using the DNA of that, why can't you use the DNA analysis of that person, knowing fully that the DNA of an individual organism is quite unique to that organism? If you want to do anything, if you want to know everything about an organism, go and you go and yes, Tajuddin Barakat, I said the full meaning of a PCR machine is a polymerase chain reaction. Polymerase for P, chains for C, and R for reaction. So that machine is what we actually make use of. So don't forget that I have only 1 minute to go. Okay. Richard Olajide, automation with instrumentation involve combining control system and measurement tools to enhance process efficiency, accuracy, and reliability. Thank you, thank you. That was a good one. I think from my end here, I can score you this. Yes, I can score you with this. So um okay. To integrate automation with instrumentation, choose instrument with compatible communication interface. Now, if you don't, you can see like USB, Ethernet, model bus, MQT, and all these are data science languages. These are program languages. That is, if you don't know them, you might find it very, very difficult to actually talk about it. It's a laboratory technique used to amplify specific DNA segment. Thank you, Taofeek Omowunmi. Yeah, to amplify, you expand, you multiply it in thousands, in cycles. You have it in cycles. You might start with just two fraction now. By the time it come again, 2 raised to power 2, 2 raised to power 3, until you have the last cycle, which is a 2 raised to power 26. So that at the end of the day, the first one that starts a reaction is going to be 2, and then after like a 1 hour, or after like at the end of the reaction, you now have 2 raised to power 26 after 26 cycle. So that is a PCR, is a laboratory technique. Huh? Technique, PCR reaction. So you don't forget that somebody that asked for it was talking about what is PCR machine. So it is a machine that they use to carry out PCR reaction. So and what is PCR reaction? A PCR reaction is a polymerase chain reaction. So polymerase is an enzyme that was gotten from a microorganism, a bacteria that we call hemolyticus, uh, hemolyticus that can survive in the presence of high temperature. So because this enzyme is a polymerase, it can survive very high temperature without being denatured. Don't forget that enzymes, they are easily denatured. They are easily denatured. That is, when you increase the any increase in temperature is bound to affect the rate or the behavior of that kind of enzyme. But with what is called that enzyme that is called polymerase, so irrespective of the temperature, even at high temperature like 96 and what have you, the that enzyme is still going to be very, very stable. So autom with instrumentation involve the use of instrument to make the process such as detection, regulation, calculator and tracking process automatic. So in short, what automation is all about is that I said it is going to make your work more efficient. It's going to make it more efficient. Automation will bring about efficiency. Going to bring about productivity. It's going to increase your knowledge about it. So once again, I want to really thank you for, especially for some of you that really ask a question in the comment section. I really want to thank you. Don't forget that the next lecture will also continue maybe tomorrow, especially microbial evolution for microbiology students. So once again, thank you and have a wonderful day ahead. Thank you.